So we are going to deal with the FACS dataset in R programming language. You need these packages such as FlowCore, Flow Workspace, CytoML, ggCyto, FlowAI, FlowStats, and eventually you can also use CytoR suite, which is which will help you to analyze your data sets very interactively. But we'll be dealing with all the options out here, and then we can analyze our data sets. And what are the steps? So first you need to collect all your FCS files from your machine, and then we can perform we can compensate it, and we can transfer the data sets. Then we go for the quality control. Then eventually we go for the gating and some visualization. If you use the CytoR suite, then you can choose your gating manually by selecting a specific region, but also you can do it automatically as well. So we'll be delving into that shortly. So yeah, see you in a bit. So let's begin. So this analysis is intended those who do not have access to FlowJo or FACS Express, those kind of commercial softwares. So this is purely in R. I mean, this is my R studio. So you can analyze flow cytometry data in our platform. Let's begin by creating a new project. I have already created. So what you can do is you can go here, and here you can type in new project, and in the okay, if I cancel it, then it will be gone. So anyway, you look into my previous tutorial on R, how to create a new project, and how you can define your project using Quarto documentation that you can find in my previous video. So yeah, basically you go here and create a new project, and there you have to input the title, the author, and yeah, these are the advanced commands that you can use if you have very large files. For instance, the FACS files are really huge. If you can see here my FACS data, you can see my data, the file size is around 200 megabytes, 143 megabytes. It's quite large. So if you have hundreds of files, then it will be more than 2 gigabytes or 4 gigabytes of data. So yeah, let's start. So this is basic introduction. You can insert images, and also you can write different types of contents in order to help you to understand what you are doing inside our studio or for your analysis. So the packages that I'm using in this tutorial, and which I highly recommend, are FlowCore, FlowStats, FlowAI, CytoML, OpenCyto, Flow Workspace, and ggCyto. These are the packages that you need to install. And also you can visit this link, FCS Express downloads, so that you can understand what you are doing. Like for instance, if you compensate, if you transform your data, how to set up the gate, and what are the statistics involved. That also you can refer to the FCS Express manual, and you can find all the information over there. CytoR suite is another package in R where you can manually manipulate the data by providing different types of information to which are your control files, which are your activation files, which are compensation files, and also you can set your gating manually inside this package. So this package is really helpful, but I'm not going to analyze by using CytoR suite, but rather I will use FlowCore, FlowStats, FlowAI, and CytoML, ggCyto, especially for the visualization purpose. And I'm going to do the analysis. So yeah, So let's start with installing the basic packages. So these are the CRAN packages that you get from the R, like tidyverse, neatar, ggplot2, remorse, bioc manager, devtools, and cable extra. You can also install grid extra. That is also the same package. So actually I have done all the analysis because it takes a lot more time. I don't want to waste by analyzing one by one. So I have installed everything, I have analyzed it, and I have created HTML file for you guys to refer to this particular tutorial. Okay, so you install the packages first. These are some of the outputs that you get, and then you go to the bioconductor and you install these bioconductor packages such as FlowCore, Workspace, OpenCyto, FlowAI, ggCyto, and CytoML. And if you want to analyze your data by using CytoExploreR R data, which I think I'm going to cover in my next tutorial, so you can install that as well. Okay, so this is the command that you install all the packages from bioconductor. Okay. The next thing that you want to do is load all your packages, and the basic command is library, and you define your library once you install all the packages. So this is the basic command that you load all the packages at once. Okay, and this particular chunk is for large files. Since FCS files, they are really huge files, so you want to read those files in chunks rather than reading the whole data sets. If you have 6 gigabytes or 4 gigabytes of data, then loading at once is very difficult. The R cannot handle it. There will be memory issues. So I think this particular chunk of command is really helpful in order to analyze your data effectively without any memory errors. Okay. The next thing that you want to do is sometimes, let me show you. Sometimes I get the files. Let me show you. Yeah, this one, for instance. You can see that these files, these are named without any underscores. I mean, there are some spaces also. Okay. Of course, these are specimen files, but sometimes you get all these files without any underscores, and there are spaces in it. So in order to avoid that, avoid the spaces in your analysis, so you can format your FCS data. And this is the particular bash command. So this is a for loop where you provide, in place of spaces, you provide the underscore. So once you do this, your data will be formatted according to, let me show you. Yeah. And it will be easy for you because these underscores have been inserted by using this bash command, so that your files would be, you can analyze it effectively without any issues with the spaces. Okay. So if you get your files with underscores and with hyphen also, that's fine. But if you don't get it, then this is the command. Okay, then what you can do is you can read single files also. For instance, if I uncomment it, the command will be the FACS data, and you load your FACS data into it. So let me just, yeah, you can see that my file has been loaded, and the command to load inside the R environment using the FlowCore package is read.fcs. I mean, this is the function that you need to load your file, and you can just execute this, and you can see that your file will be loaded. Okay, let me just load all the packages, then I think it will work. Yes. So now since I have loaded all the packages, now if you execute this particular command, you see the read.fcs is the function from the FlowCore package. So this will be loaded, and you can analyze all the names in the FCS files, even the expressions or the keywords, the metadata, etc., by using this particular command. So names, you can see from the names you have FSCA, FSCH, SSC, side scatter, forward scatter, and these are some of the markers. I also described all the abbreviations, what they really mean, so that you can also refer to that documentation. These are the expression values that you can get from this particular single file, and you can see these are the expression values, and the command is exprs, and you get all the expression values from here. Okay, and if you want to analyze what is the median value of all your expression values, you can use this command to find out the median values, and these are the median values, as you can see, per sample. Okay, I mean for your sample that you have loaded. And this is the command to extract the keywords. You can see that what type of keywords has been used, and if you can open the FCS file also by using text editor, you find the similar information. But in R it's much more easy, so that you write one command keyword your FCS file, and that's it, you get all your metadata over here. Okay, so this is how you can analyze a single file. If you want to read the whole data set, again this read.flowset command is really helpful. This will load all the files. I have 5 different files, FCS files, so you can load all the files at once. Truncate max range dot names is equal to true. That means you can print out all the values inside the files by using this. Otherwise, if you keep it false, then only half of the files will be displayed. Otherwise, if you set it for true, then all the values will be displayed. So if you execute this command, you will see that your so I give fs as flowset. That means all the files in your data folder will be loaded to this particular variable, which is fs, that is the flow set. Okay, now it's loaded, and if you want to analyze a single data set, you can see that it will tell you that a flow set with one experiment has been loaded. If you click on fs, then you can see that a flow set with 5 experiments. You can see I have 5 different files, so 5 experiments have been loaded. If you want to analyze the column names, then it can tell you what type of column names do you have. So starts from forward scatter to all the markers, side scatter, time, and all types of different markers are there. Okay, and if you want to extract the metadata from this, there is a function called pdata that you can execute. So for instance, if I execute this, it will tell you what type of files are there, which FCS files have been loaded over here. And for instance, if you want to extract, if you want to specify this with a naming scheme, for instance, here you can see that this pattern consists of different types of dates, right? So what I can do is I can extract these dates and put it in a separate column called well, or maybe date, whatever you wish that you can insert the name. For instance, I put well over here, and this is the expression, or the expression to extract these dates and put it in a separate column. So if you execute this particular command, you will see that, and then this particular command, you will see that a new column has been created with well, and you can see that dates have been inserted into the well column, so that I can specify the sample name with these particular dates. And while plotting also, it will be helpful so that you recognize all your samples. If you specify this, then this might be a little bit too much for the plot. So that's why you can separate, for instance, in my case it's the date, but you can do from different markers or different wells or whatever you like. I mean, how do you want to specify the naming of the samples? It depends on you. Okay, and if you analyze the column names, so you find, yeah, these are the column names actually, and this is a very nice command, that is the parameters data. So if you click this one for the first experiment, you can see that you have 33 different parameters. So if you click this particular thing over here, it will open a new window, and you can see that you have 33 different parameters, as you can see, including the time and your forward scatter and side scatter values, and these are your markers. And you can see in the description that these particular markers are CD24, IGD, CD56, CD38, blah blah, you know, all these markers are there, and this is the time. So whatever the colored markers are, those are having some description, but with the forward scatter and the side scatter, you only and for the time you don't have any description over here. Okay, that's normal, right? So this is how you extract the metadata from your samples, and the abbreviations used as I mentioned, forward scatter, H means height and A means area, similar to the side scatter also. And you have FITC, that is fluorescein isothiocyanate, and this is a fluorescent dye. And also you have all different types of fluorescent dyes I have written. For instance, BV is a brilliant violet. And the time, this usually refers to the time parameter, indicating the duration of the cell's passage through the laser beam, which can be useful for identifying doublets or aggregates. Okay, so be careful with all these parameters. If you want to change the namings also, you have the option to change the names. For instance, if you, so I commented it out because I don't want to change it, but if you look into this, so what you have to do is you have to extract the column names. And for instance, if you are looking for this BB660PA, you can refer from here. Okay, let me execute this command, and so it's BB660PA, that is CD56. So what you can do is you can go down and you can change it to CD56. Okay, I think this is changing because I changed the font, so that's why it is bit overlapping for the cursor, so it doesn't matter. So you can change the namings like this, you know, the column names, and you can change the naming patterns from here if you wish. Otherwise, leave it like this. You can see FSCA is FSC, and if I want to change this particular marker to this particular name, then you can change the namings. I mean, it depends on you whether you want to change or not. Otherwise, leave it for now. Okay, the next thing, and the most important thing, is compensate your sample manually. I mean, you can do it automatically also. There is a command to do automatic compensation, but I would recommend to do it manually in order to understand what are your fluorescent dyes that you have used, what are your controls, what are your activation samples. In that case, you can use particular sample. For instance, a control sample that you have, you can compensate your samples manually to the other samples. Okay, so basically what we do is we create a spillover spreading matrix. I think most of you, those who are using Flow Cytometry, the FACS Express or FlowJo, they have this particular function which you can do it manually and automatically as well. In R also you can do the same by creating a spillover spreading matrix. Okay, and I have written all the description over here so that you can read. So I'm taking the first sample as the control sample, and on the basis of that I am compensating all the samples. Okay, so the command is spillover fs, the first sample. So if you click here, you can see that your samples have been, it will create a spillover matrix, and then by using this command compensate, this is the function, so compensate command, then you can compensate all your samples. It will take some time based on your on how many samples that you have, but yeah, it will be done quickly. And then if you check the parameters data, you will see your maximum range, minimum range, and the description, and your naming with all the parameters like this. Okay, so this is how you compensate your samples. Here I am taking the specimen sample, so don't bother about that. If you have the real data from the flow cytometry, then you can do much more better. The next thing that you perform is the quality check. This particular thing will take a lot of time based on your samples. Since I have samples, almost maybe 1 gigabyte or something, it takes a lot of time. Probably for yesterday I was doing 10 samples, so it took almost half an hour to do the control and the quality check. And the command to check the quality of the samples is flow auto QC. It will automatically do the QC for you, and it will produce a QC folder like this, results QC. Per sample it will create QC, the quality control files, and also an HTML file, so you can view it in your browser. For instance, this one is called Flow AI report. So this is the flow, the quality check is the package from Flow AI. So this will generate a report for you, and you can see quality control analysis, the summary, the how many anomalies were there, how many anomalies were detected, number of high quality events, flow rate check. Okay, so signal acquisition check, etc. So you can find all the information over here with all the parameters. You can see from here to here, all the parameters have been listed here, and you can see what type of change points detected in the channel shifts with the dynamic range check, negative outliers, upper margin events, and anomalies also. Okay, so this is a very nice report from here. For each and every sample, you can view all this quality report from by using the HTML file generated over here, right? So yeah, so this will create a folder called the results QC, and also it will give you a mini.txt, I think. Yeah, you can see how many anomalies detected here, the text for the metadata file, for instance, for quality check. Okay, so this is the file that you look into, right? I'm not going to run this because it will take a lot more time. The next important step is the transformation. Transforming the flow cytometry data after the compensation is a very important step in the analysis process. So what you want to do is you want to use this function called estimate logical. So this will compensate, this will transform your samples based on the parameters that you provided. For instance, 5 to 32 means my samples, if you look into here, if I click this, and you can see I want to transform all the samples from parameter 5 until 32, not the time. Okay, all these markers, and maybe these are my control samples, so these are the dice that I want to transform these samples. So I input parameter number 5 to 32 here, 32. So as you can see here, I have input this 32. So if I press this, so it will generate a transformed matrix. Okay, now if you I give a variable name such as fs comp clean fs I mean the flow set compensate clean transformation. Okay, and then I transform all the values. If you look into the first experiment or your first sample, it will take some time to transform all the values, but it will be done quicker. I mean, at first I mentioned that R can handle large number of files. In FlowJo, if you, or maybe in FACS Express, if you load many samples like 100 or 200 samples, then it might break. Sometimes it will cause problem, but in R it can be done effectively. Okay, now it's done. So if you look into the first experiment, you see the values over here. You have all the 33 parameters, and you can see dynamic range, your minimum range, your maximum range, etc. Right, and you can look into the parameters data as well here, right? Okay, so this is how you transform all the values. Then you visualize the results using the ggCyto package. This is one of the most important package for flow cytometry data analysis, and the function that it uses is autoplot. You don't have to use ggplot2 or maybe any other package to plot the data. Autoplot can handle everything by its own. So it's just the basic command autoplot, and then you insert your sample or your experiments, and that's it. It can produce very nice plots. So if I click on this autoplot, you can see that a very nice plot will be generated. Yeah, it will take some time, and you can see that this is before the transformation. I think, yes, this is before the transformation. So that's why I mentioned that transformation is why transformation is necessary. You can see it from here. So this is before the transformation, and you can see this one plot after the transformation. You can find the plots much more informative in respect to the file which is not transformed. Yeah, look at this plot and look at the previous plot. It's really different, and you can find much more information from this particular plot. Okay, so that's why transformation is really necessary, right? And then if you want to plot your forward scatter with the side scatter, you can just use autoplot function again. Our plot has been characterized by using the well information, that is the date that I have previously generated, a column called well in that date. So you have your specimens or the experiments well characterized, so that you can identify all your samples. You know. Then you have visualization with other markers. If you want to visualize with other markers also, like forward scatter, and this is one type of marker, then you can find different plots in order to analyze. I mean, these are all specimen samples, so don't bother about the plots. It might be correct or it might be wrong. And then against time also you can plot these plots. Okay, the next important thing is the gating set. Now I gave the explanation over here why gating in flow cytometry is really important in order to identify the cell population, the singlets, the doublets, and live cell, dead cell. You do the analysis in flow cytometry, right? So you in the gating set, it is very important that you transform your data and insert that particular transformed object into the gating set. Okay, so this is the command to execute, for instance gs, and your gating set is the command, and then your transformed object over here. It will take some time based on your samples, but it will be done quickly if you have less samples. Okay, you can read all the documentations over here. I don't want to go into explaining why it is important. So I have written it over here very nicely so that you can understand all those things. I just want to quickly show you the codes that are used to analyze all the flow cytometry data. Okay. Next, you want to define the cell gating. So the command to define the cell gating is gs-pop get data. This is the new command. If you follow previous tutorials, like the old commands, then it will be only get data and then your matrix. So I don't recommend, or your gating set. So I think this command has been changed recently. So this is the new command that you can define your cell gate. Okay, so if you click this, you can see you have defined your cell gating, and I want to define this particular cell gating into a variable called myGate. So then this is the command to insert gating into your gating set. Okay, so I name it as myGate, and the parent is the root, and I gave it a name called myGate. Okay, then you have this particular function called gs-pop-add. So this will add the gate into the samples, and then you recompute it, and you can see that it will take some time to apply the gating. And you can see it over here. I have a for loop plot for all the samples for forward scatter and side scatter, you can see, and then you can print out all the gates over here. So it will take some time, and it is using FlowClust. I think this is from one of the packages that we installed from Bioconductor, and you can get all the information from here. Why the cell population, why you are doing for singlets. Okay, all these things that you get all the information from here. I mean, you are using commercial software, so basically sometimes it is very difficult to understand what these commercial softwares they do. Actually, it's kind of a black box. Even though they have very nice manual, nobody bothers to read it. So maybe it's better to try it once in R and then go for commercial softwares in order to do the analysis so that you understand what you are doing. Yeah, also I want to show you that this is my R repository. GitHub repository. You can refer here. I will insert all the codes over here so that you can refer from here, and also the HTML file that I have generated from Quarto document, and also all the codes in here. I don't want to upload the files because the files are too large, so you can analyze from your files, but I will upload all the codes over here. And also the explanation is inside the code, so I don't want to write it over here, but I will upload all the codes that has been generated, and also the QC files also, but not the bigger files, but also these smaller HTML files I can upload. Yeah, it's done. Okay. Here I mentioned recompute, so that's why it applies the gate, and then it added this particular name myGate into the samples, and then recompute. So that's why twice it has been executed. Okay, now if you click here, you can find how the plots are being generated and how the plot will look like. For instance, if you go here, yeah, this is one plot, this is another plot here, this is another one, this is another one. Yeah, you can see. Okay, and then you have to input the singlet get. Okay, it's the same. You refer all the information that you have defined over here, that is the my gate, from here or here, the my gate name, you have to insert it over here. You have to use the gating set and also the same command from here. Okay, so now you define the singlet gate. You have to define your singlet gate, because this one already I have defined over here. Okay, so yeah, it's done. Next, add the singlets, and then a recompute. Done. And then auto plot. You can see this is my auto plot diagram here, and you can see that all the samples, the singlets has been defined over here. This may not be accurate, I don't doubt that, because these are specimen samples and this is only for the tutorial purpose, so don't bother about that. But if you have real experimental samples, then that would be really nice to look into that. Okay, and you define your samples from here. The plotting, you see how it has been. I mean, this is automatic thing, so maybe you can go for the manual gating also. That is also another thing, that you can define your gating by using different as I mentioned, CytoExploreR suite, you can define your gating, but this is automatic one, so it might predict accurately or precisely, or may not predict accurate enough, so you can go for manual gating also. The quad gate is optional. If you want to define your quadrant, then also you can define by using this particular code, and then you perform then you go for the statistics part. So gs popset stats, this is the command that generally we use to get all the statistical values. Okay, so I just execute this command for your gating set, and you can see that all your counts has been written over here with all the population, that is your root, your mygate, your mygate and single. The mygate is the thing that we defined previously, and you have the root samples, and you can define your singlets as well. Okay, so the next thing is your mygate and percent that you want to get here. You can see your percentage as well. Okay, and then you have your with the population, you can see along with the populations you have your gate, your channels, and you get to do all the statistics I think based on your samples. So this is just an example. And then the next thing is you define a function, and then get the quantiles value, like I think 75% quantiles. It will take some time in order to generate, and all the information that you need is over here. Why I am doing that, and what is the importance of this, and also statistics is really important parameter in order to understand what are the values really mean. For instance, if you go here for MFI is a measure of the average fluorescent intensity of a cell population, so or the mean fluorescent intensity, MFI. I think this is the most important parameter in flow cytometry analysis, right? And pop quantiles calculates the 75th percentile, as I mentioned, or any other specified quantile for each channel. Okay, you can read the documentation, and you can see from here, I think, so all these values will be generated, statistical parameters. Okay, Now you have another function where you can define your mean values. So this is pop mean, this is quantiles, this is the mean values. And also the next thing, yeah, now you can see here you have all your mean values. Okay. Also the next thing is get the population statistics for the downstream analysis, which is the gspop get count with a meta function. This function you can use it by using a variable called ps, and then you define your count and parent count, and this is the function, and you can generate a table. Actually, you can see here a table with all the information, all your samples, the well information or the naming scheme that you have defined, what is the population, what are the counts, what is the parent count, and percent of the parent. Okay, so like this you can perform your flow cytometry data analysis in R, and this code will be publicly available, so don't worry, you can use this code for your analysis purpose. And if you want to save the whole session of the R file, then you can use this command save image and your R data file, and you can also take a snapshot of your R environment that all the packages have been loaded, so that when you move to other laptops or any other, if you want to send it to your colleagues or professors, then they can utilize the same R environment and can install all the packages that you have used and reproduce your data. Okay, so if you click on a render over here, if you are using Quarto document, it will generate a HTML file like this. It will take some time to generate the HTML file. So now you can see that HTML file has been generated. Here the packages and the outputs also I mentioned here, and these are the codes. Okay, and these are the plots also I have given with all the explanation, so that you can refer to the documentation itself. You can see. Okay, and then you can use it for your own analysis. I have two different HTML files. So one is this one is the previous one as I have shown, and here you can see, if you click on show all code, view source, here you get all the codes like this. You know, you can directly copy paste from here, or if you want to read it like this, and you can just unfold and read. Also look into the execution of the packages. For instance, if you go here, yeah, this is the code, and you can see flow set with 1 experiment because this is the parameter I have used, and then fs flow set with 5 experiments. Okay, so you can actually look into the code and then look into the outputs as well. So I will upload this to this particular GitHub channel. I will put the link in the description, and I think you can refer that. Okay, that's it for now. Maybe I can later on, and yeah, also forgot to mention that this is a CytoExploreR package in which you can actually manually get and define this type of cool plots, but the important thing is you need to have two sets of data, which is compensation samples and your activation samples. This is really important that you categorize your samples into two categories, then only this will work. Otherwise, no. Otherwise, I think you can go for the other code that I have shown. It's a very cool package. I think I will cover it in my next tutorial with the example datasets from here only, so that you can understand. But for now, if you want to refer, then you can refer to CytoExploreR package. Okay, so that's it for now. Thank you very much, and hope to see you in the next tutorial. Please subscribe to my channel if you like it. Thank you very much. In the previous video, we have seen how to analyze the flow cytometry data using R. Today we are going to explore the CytoExploreR package, which was previously known as CytoSuiteR. So this has been changed by the author to this particular name, and this is the complete workflow how you can do gating, manual gating, and also you can explore the automatic gating as well. But this package has some more features that you might expect from a commercial software. Those who do not have the access to the commercial softwares, they can explore CytoExploreR and utilize the functions that has been implemented by this particular package. First thing that we are going to do is load all the different packages. I mentioned last time in my previous video how to load all the packages that have been listed over here. And you save the flow cytometry files into two categories. One is activation, one is compensation. So compensation is used for the controls and activations are the real or actual samples. So diving into the workflow, first, we'll do the compensation of the control samples. We prepare the compensation controls. We use different types of commands such as cytosetup, and we transform the fluorescent channels by using spillover matrix. And then we perform the gating for cells and also for the single cells as well. Then we visualize the compensation controls by using a function called cytoplot. Okay, this is all about compensation control samples, but when you look into the activation samples where the real treated samples are there, so the first thing that you do is load the activation samples, annotate the samples based on different type of fluorescent dyes that you have used, apply the compensation. Basically they use logical transformation, which is the default one. Then you perform the gating for the cells and the single cells. And then we dive into the visualization or gating where you can manually gate all the samples to categorize live cells, dead cells, CD8, CD4, CD69, T cells, dendritic cells. Okay, and finally we are going to explore the gated samples using gating tree. So it produces a tree kind of a schema or a tree kind of structure where you can see what you have done to your activation samples, and then you can explore all the statistics by using cytostats compute. So yeah, this is about the workflow of CytoExploreR. Let's dive into the R environment where you load all the packages. So I have prepared an R script. You can see from here. So the first thing that you do in order to explore the CytoExploreR package is to set the working directory, because this is really important. If you mess up using different directories, some base directories or maybe some other locations, then it might not work. So I would recommend everyone to initialize a new project and then make a folder called CytoExploreR or whatever, FACS analysis, etc. And then set your working directory. If you want to initialize the previous environment, then you can use renv init function. So this function will load all the packages or install all the packages from the previous environment. Then you can increase the memory and use the cache for the larger files. I think FCS files are too large, so I would recommend everyone to use this function to increase the memory and use the cache for the larger files. Okay. For the graphics rendering, I would recommend everyone to install OpenBLAS and LAPACK. So you can do via brew install OpenBLAS and brew install LAPACK, which is available for Mac. For Windows users, I think there are certain commands that you can install these packages and then export these LD flags and CPP flags. After installing these packages, you will be redirected to these particular commands. You just export that and then you start installing all the packages. So these packages we have installed previously in our flow cytometry analysis video. Tidyverse, neathard, ggplot2, etc. And then you install all the bioconductor packages such as CytoML, ggcyto, FlowAI, FlowWorkspace. And finally, we dive into the CytoExploreR, which you can install it from GitHub with these particular commands. So all these codes I have included in my GitHub account. So you can find the CytoExploreR.R script. So if you click on that script, all the everything has been written over here. You just copy paste and then you start analyzing your samples. To demonstrate the features of CytoExploreR, we'll also need to load CytoExploreR data, which we have installed by using this particular command. that contains both the compensation controls and your treated samples as well. So first thing that you have to do is you have to load all the required packages by selecting all of them and then command enter. So all the packages will be loaded into your R workspace. Now you should create a folder called CytoExploreR or FACS Data Analysis. So in that, if you use this particular function called CytoSave, you can use the data sets from the CytoExploreR data and save those samples as compensation samples. So I have already saved over here, but let me also click this command. So here it is written that files will be overwritten in the compensation sample. I said yes. Now you can see when you open the folder compensation samples, all the compensation controls have been loaded over here. Similar way, you save all the activation samples or the treated samples. In that, a folder will be created called activation samples, and all the samples have been loaded over here, right? Next, we do the compensation of the fluorescence spillover. What we can do here, as I mentioned in the workflow, that we prepare the compensation controls, and then we create a CSV file called compensation gating template.csv, in which when we gate our samples to the number of cells and number of single cells, all those gating platform will be recorded into this particular CSV file. So if you click this, a file will be created in which we have the metadata. If I enlarge it, we have the metadata called experiment markers editor. So this is the default one which has been provided by the package itself, but you can mark all your samples accordingly. For instance, Alexa Fluoro 488A is having CD8 marker or CD69 markers for this particular channel. So these are the different channels, and these are different markers. Now you click save and close, and if you close this and open the RStudio, again another viewer window will be open up, that is experimental details editor, in which these are the name of your compensation controls. Okay, now you click save and close. Now it's done. So it has created a file over here. This is an empty file, empty CSV file. So when you perform the gating, all the gating population will be recorded into this particular CSV file. Okay. And remember, I have previously used these abbreviations, so you can read about all the abbreviations that has been used. The next thing that you want to perform is transform your fluorescent channels. The default is the logical transformations. So this has been done by using the function called CytoTransform. So I do this, and you can see that all the transformation has been done. If I can zoom it, you can see how the transformation has been done with all the different channels, and these are the percentage of mode. Now we perform the gate cells. This is one of the feature. Those who do not have any access to the commercial softwares, they can use this particular package so that they can draw different types of gates manually, and they can utilize this for analyzing their FACS data samples. Okay, so we parse the variable that we have transformed all the samples into, and the function that we use here is CytoGateDraw, and we name it as cells, and these are the root. The parent is the root, and these are the channels, and the parent is the root. Means in the later section, what we are going to do is we are going to create a schema or a tree. In that you will find out what this root means. So from where we started and where we end, we can explore that in that particular tree schema. Okay, the channels that I'm using now is forward scatter and the side scatter. So these are the control samples. So if you click this, our session will be opened over here. Then using this particular pointer, you can click here, and you can create a polygon actually, basically. here, here, and then end it to the point that you have started, and then press right click, and here it will be written total number of cells is 66.48%, right? Then you close it. So this is a combined events, or all the events, or total number of cells population in forward scatter and side scatter region. So now all the cell population has been done. You have done the gating, and now you perform the gating for the single cells. So if you click this and again open the R session, so you can find all the single cells over here. So you can plot like this, and over here, here, and then click to the endpoint, and then you right click on that particular point, and you will see my single cells are 98.56%. So it depends on your skills, that it comes with the practice, that how well you can define the gate. But this is just an example, so don't worry about the difference or the variations that I'm getting here. But in your experiment, you might consider it very carefully. So with practice, it will be perfect. Different points to consider: that whatever the channels that we have used, you should know about the definitions of that. For instance, Alexa Fluoro 488A is a green fluorescent dye, often used to label the antibodies, peptides, or proteins. Similarly, I have explained whatever the channels that I have used in the example data set. The next thing that you do is compute the spillover matrix. So it creates a spillover matrix.csv file, and the parent will be your single cells. Remember, you have done both for all the cells and for the single cells. So this spillover matrix is only for the single cells. So once you click this, a viewer editor will be opened up, and you can define your channels over here. So I have described the example data set, what channels that you should select. For the compensation 7AAD, 7AAD is typically excited by a red laser, and you can select the channels by using these particular channels. So 7AAD we can select from here. Okay. For the next thing, AF700, we choose Alexa Fluoro 700A. Alexa Fluoro 700A. For the APC-CY7, you choose APC-CY7A. APC-CY7A. APC is typically detected in a channel similar to Alexa Fluoro 647. So Alexa Fluoro 647, we can choose yes. And then for the FITC, Alexa Fluoro 488, because FITC and Alexa Fluoro 488 have similar excitations. So 488. Compensation for the PEA, as this channel is intended for the detection of the PE dye. So PE, I think, PEA, yes, PEA. And then your unstained one is your unstained samples. Okay, and now you save and close. Now here it will be written select a fluorescent channel for each of the samples. So selecting the lower and the upper bounds of the 7AAD, I mean, it will pop up. So when you open the R session, you can see all your compensation graphs. So in order to properly compute the spillover values, CytoExploreR requires an appropriate unstained reference population. This reference population can take the form of a universal unstained control or unstained events with each compensation control. So when using universal unstained control, the unstained control will be overlaid onto the plot, and users be asked to get the positive signal for each of the compensation controls. Otherwise, users will be asked to get the negative and then the positive signal for each compensation controls. The accuracy of this particular spillover calculation is heavily dependent on where these gates are positioned. With practice, it will become clear that is best to retain the final peak and tail in the positive gates. If you go to the files and then open up the spillover matrix, you can see the values have been written over here, right? So this is your spillover matrix. So it depends on what you choose, what type of, or whether you choose the negative compensation or the positive one. So yeah, you can read about the documentation how you can choose different channels by using the gating command. And then when you click on the variable spill, you can see the matrix over here, right? Now once you have done the spillover matrix, you can edit the spillover matrix dynamically by using this command called CytoExploreR edit. If you have done something wrong, then you can actually look into this particular command and you can edit all your spillover matrix samples. For instance, you can see all the values have been written over here, and these plots will be created if you have installed the OpenBlast and LA pack, which I have mentioned in the beginning. So you have to be I mean, for the graphics rendering, the BLAST package is really necessary, otherwise it will throw an error. Okay, so you can compare all the samples. For instance, for AF700 compared with the unstale control, and then you can select all the samples and all the plots will be generated. This is the density plot. Okay, and you can select whatever you like this. You can edit, even you can input the values over here that you have. And if you go to the plot section, you have all the plots generated, and these plots are really helpful to analyze each and every compensation channels. For instance, if you go here, it will be changed, and you can explore these plots or save these plots if you need, and you can use it as an supplementary material for your publication as well, right? So yeah, this is how you edit the spillover matrix, and this is the editor by using this particular command that is called CytoExploreR edit. Okay, now you visualize, actually visualize the compensation controls that you have previously gated and parsed into the spillover matrix. So if I click this, so if you go to the plots and zoom in, you can see all the plots will be automatically generated that you have seen in your spillover matrix editor, and it will show up all the populations, so it will be listed as CD697AAD because that's the marker that you have used for that particular channel. So it will generate all the plots. If you require all these plots, then you can add into your supplementary file. Otherwise, just for your analysis, you can just keep it as it is. It will take some time, but all the plots will be generated. So after visualizing the compensation, you can actually parse the spillover matrix and again look into your compensation sample. Please check out my previous video regarding the compensation of control samples and how to calculate the spillover matrix, and also how you can manually gate the samples for the total population of the samples and also single cells as well. Okay, so once we have finalized the spillover matrix, we are done with our compensation controls. And now we can move on to analyze our activation samples. The first step is to load the activation samples, annotate the samples based upon what samples you have in your experiment, apply the compensation, again the logical transformation. Then we do the gating for the total population of the cells and single cells. And finally, we are going to visualize the live cell, dead cell analysis, even the markers like CD8, CD4, CD69, T cells, dendritic cells, based on different gating patterns like rectangular, polygon, circle, ellipsoid, etc. And finally, we are going to explore the gated samples using the gating tree. I mentioned in my previous video that we can also get a schema that you have done in your analysis, starting from the control samples to the activation samples. And also you can plot a gating tree. And finally, we are going to explore the statistics by using some functions of CytoExploreR. You can start by setting up all your activation samples. And the first command that you are going to implement here is that you are going to pass a variable called gs, which you have previously done over here for the compensation controls. Now we can analyze the samples and load and annotate the activation samples by using the cyto setup command. And if I run this command, so you can see that your activation samples have been loaded, and it can show you what type of markers you are using and what kind of the channels that you're using. So we are using the default one because we are using the data sets from CytoExploreR data, but based on your experiment you can choose the markers. Save and close. And you can see the experimental details editor, where you can see there are 33 activation samples. So you can annotate them to easily identify what samples they are. But for the sake of this tutorial, I'm using the default data sets. Okay, save and close. Now, activation gating template has been created, as you can see over here. Yeah, activation gating template. This is similar to the compensation controls gating template that we previously created over here, this one. And now we have created for our samples. Okay. If you have messed up, something, you have done something wrong in that, then there is also a command called clean is equal to true. So that can help you to edit your files. Next, apply the compensation, and the default is the logical transformation. So we create a spillover matrix.csv. Now I'm going to create. Yeah. So it will be created. So now the spillover matrix has been created over here, and you apply the logical transformation to all your samples. Since the data is always imported uncompensated and on a linear scale, we'll need to apply our spillover matrix and transform the fluorescent channels to appropriately visualize the data. As you can see over here, now since you apply your logical transformation, you can actually visualize your data. Otherwise, it would have been in the linear scale, and that would be very difficult to visualize the samples. Next, move on to gate the cells, the total population of the cells in the samples. So again, the same command for the forward scatter and side scatter we are going to use. This is for activation samples. Remember, this is not for the control, but activation samples. So just left click. Okay, so I'm taking a big chunk of population over here, and yeah, let's see, and then right click, and you can see the total population of the cells is 64.56. so you are covering almost 64.56% of cells. Now it's done. Next, you can apply for the single cells. Now click that and open the R session, and now you can see your single cells. So what you can do is you can start from here and then go up until here and over here, and right click, and you can see the total population of the single cells is 96.26%. Okay, and now what you can do is you can create a variable called nil, and you extract the unstained controls, because previously we have seen that we have marked our experimental editor in which we gave a channel name called unstained controls. So you can extract those unstained controls and retain all other samples. So that you can do over here. This is applied to 33 samples that we have seen, that we have total number of activation samples is equal to 33. So you apply to all the 33 samples, so that's why there is a number 33 over here. Okay, you extract, now it's done, and if you type in nil, so you will see how many unstained controls have been extracted from that. Okay, next we are going to analyze the live cells and dead cells. So it's called live cell dead cells analysis. So this is a very common analysis in flow cytometry data. So I would recommend everyone to use this particular function, CytoGateDraw, so that you can analyze the live cells and the dead cells. And the type that you are using is a rectangle. So I'll show you how to do that, and open the R session. So now you can see that you have the population of here the live cells, and these are some of the dead cells. So what you have to do is, for the rectangle, you click the diagonals points, this particular portion, and you use this particular point, and you can see these are the live cells and these are the dead cells. So this is how you can annotate the live cells and the dead cells. So dead cells percentage is around 42.69 and live cells are 57.31. So yeah, don't worry about the results, these are not accurate, this is just for an example. So this is how you can do the live cell and dead cells analysis. Okay, for the rectangle, tap the two diagonal positions and it can show you the population. If you have done something wrong, then you can edit the template. So for that, cyto gating template underscore edit is the function. You can use it. So to gate the T cells and the dendritic cells, we use the CytoGateDraw function, and the type that we are going to use is ellipsoid for the T cells and rectangle for the dendritic cells. Okay, so once you click this command, so first you have to do the ellipse for the T cells identification and rectangle for the dendritic cells. So that you can do by opening up the R session. Then first we have to do the T cells. These are the T cells. So you have to click on 4 different points. So point number 1, point number 2, point number 3, and point number 4. So it will tell you the T cell population. This is how you can draw the ellipsoid. By clicking 4 different points. So T cells is 34.59, and the next is dendritic cells that you can use a rectangle. So click here and this point, and it will tell you the dendritic cells. So the percentage is 7.31%. We have seen that CytoGateDraw has a support for the mixed gate population, that is ellipsoid and rectangle. Now we are going to gate the CD4 and CD8 T cells. So we are using both for the CD4 and the CD8 T cells the rectangle type gate. So if you click here and open the R session, you can plot 2 diagonal points like this, and this is your CD4 T cells, that is 33.83%. And for the CD8 T cells, we'll see how much it's around 50.71. Okay, so this is how you can plot CD8 T cells and CD4 T cells by using the rectangle gates. You can create a variable called CD4 and extract all the CD4 T cells. So now the CD4 T cells has been extracted. If you click, and you can see a cytoset with 33 samples have been documented into the gating template. After extracting the CD4 T cells into the CD4 variable, we can now gate the CD69 plus and CD4 T cells by using this, and we are using the rectangle type. So first the channel cells CD44 and CD69. Okay, now if you click this, So you have this very nice overlay. So these are your CD69 and CD4 T cells, so that you can plot by using a rectangle. So for instance, I take this point and maybe over this point, and you can see that your percentage of CD69 plus and CD4 T cells is 2.41%. Okay, now we are going to get CD69 and CD8 T cells. That also we can get, and also we have this particular command called contour lines in this particular function. So you can check out all whatever the commands that are available for CytoGate draw by using question mark and then paste the command, and you can see what different types of parameters available for this particular command. So if we click here and let's see how it looks, and you can see a very nice contour lines. So we gave only 15 contour lines, so it was overlaid into this particular plot, and now you can plot your CD69 plus and CD8 T cells by using the rectangle. So you click here or over here, so it's around 1.15%. Again, these values are not accurate. It's totally depends on you know your experiment better. The next thing that I mentioned previously also, we are going to use a schema or a tree so that you can understand what you have done previously, starting from the compensation controls to activation samples. This cytoplot gating tree plots the gating tree with nodes scaled by population size, and can also display the frequency and count statistics for each node. So it's a better representation of what you have done until date. So stats, that is the frequency, and if you click this, so it will present a very nice let me zoom it, a very nice schema or a tree, so that you can see what is the percentage of the gating parameters that you have obtained. So let me make it bigger. Yeah, you can see the dead cells is 22.51%. The root is your where you have started, the parent root, and cells, total number of cells is 64.43. Then single cells is 95.93. Then you have categorized the live cells and dead cells from here. Then you have identified the dendritic cells, the T cells, the CD8 T cells, the CD69 CD8 T cells, CD4 CD69. So it's a very nice representation of the diagram that you have done until that. If you find something wrong in your experiment or in your calculations, then you can again start from the beginning itself. Okay, so yeah, this is about the CytoGate tree. So let's dive into the schema, how the schema will look like. So it will take some time, I think. Yeah, this is the plots. So if you can zoom it, so whatever you have done until date or until now, that you can identify from this. So the first thing that you have done is cells, then single cells, then you have categorized the live cells and the dead cells, then T cells, dendritic cells, CD8 T cells, CD4 T cells, CD69 CD8 T cells, and CD69 CD4 T cells. So again, it will give you a very nice representation of your events. Okay, and finally what we are going to do is we are going to compute the statistics by using this function called cytostats compute. And if you click here, so it will give you a very nice statistics analysis. So it will present a table. You can save the table as well, but these are your statistics. It computes the desired statistics for the specified populations and return a tidyverse friendly output that retains all the experimental details. These are some of the statistics that you get from your analysis point of view. The one thing that I like the most is about this particular tree, so that you can give it as a publication figure. So yeah, that's it for the activation samples. Later on we can dive into all other packages that are available via bioconductor or any other Python package I will try to explore. Or please comment below that if you have any suggestion for me to explore or deep dive into flow cytometry analysis using R or Python. And please subscribe to my channel, and thank you very much.